Welcome to The Edge, a program where we spotlight Purdue University faculty, scholars, and innovators, highlight their careers, scientific achievements, and their vision for the future of their scientific field. My name is Tommy Soares. I'm Assistant Director of the Purdue Institute of Inflammation, Immunology and Infectious Disease. And on this segment, we continue focusing on microbiome sciences. I have the pleasure of welcoming Dr. Roland Wilhelm, who is Assistant Professor in Environmental Microbiology in the Department of Agronomy. Welcome, Roly. Thank you so much for being on The Edge today and sharing some of your insights and knowledge with us. It's so great to have you on the program.
Yeah, no, I appreciate you having me, and thanks for the introduction. Yeah, I'm excited to talk about what
I'm excited to hear about what you do. And I'm excited to hear a little bit about the story because I know that our paths even crossed. I see that you did a stint at the University of Guelph, which I also did. I did my undergrad and master's there.
Oh, really?
Yeah. So, and I'm Canadian as well. But, I just want to hear this whole journey, this whole adventure of how did he get to Purdue University? And I see that you have a very rich path and a rich past of, microbial ecology and soil microbiology. And you've been really focused on this field for so long. And I'm so excited to hear what the journey is all about. So take us back if you can.
Yeah, well, if you can believe it, my father, let's start with the environs where I came to know the world, right? So my dad was on a ship he moved cargo up and down the Great Lakes. So on our vacations, he was away from home a lot. That's the life of a sailor. So on our breaks and vacations, we'd go and join him on the ship and go up and down the waterways and have this amazing, it was kind of boring, but it was also kind of spectacular because you're on this big hulking boat that's essentially an engine you can walk into. The engine of that room is so big that you can walk in and see all the different parts of an engine. And then we had all these binoculars. So I'm not surprised I ended up being a microbiologist because I would spend my time just staring at the horizon, looking for wildlife and all sorts of stuff. So that's what started me off on my journey into sort of environmental interest in the environment. And I actually started as a marine biologist in my undergrad. It turns out that I couldn't cut it when it came to mammalian physiology. I'm faint of the heart. And so I almost fainted a few times when I was doing all these dissections and stuff. So I had to quit. And so I transferred into ecology. And then I realized ecology didn't have all the juicy stuff that I really liked about science, and that was chemistry and biology. So ecology, in its purest form is really math. And that's okay, but I missed the wet lab component. I missed growing things and working with things. So I found the sweet spot where I studied environmental toxicology, which is essentially how chemicals, typically human generated chemicals move through the environment, how they're changed through the environment, and whether they whether they have detrimental impacts on the life of the environment. And so it's a fascinating topic, actually. I really recommend people look at that if you find yourself in the environmental sciences, but you also really like the hard sciences, STEM, chemistry, biology, math, because these are complicated questions, right? There's a lot of different processes that happen out there that you have to be familiar with and learn how to study. But it turns out that the career in environmental toxicology leads you really towards either the government, where you regulate certain chemicals and that we introduce in the environment, or into the companies that produce them. And when I came to that sort of fork in the road, I wasn't really excited about either of those opportunities. I was actually drawn by a desire to do more research. And also I thought I would be a part-time science journalist. So I had actually done a degree during my undergrad. I did a minor in English, and then eventually I completed it for a full bachelor's of English. So that is atypical. And it got me into all sorts of trouble too down the line. When I was applying to grants and stuff, everyone's like, what's this English degree for? So yeah, that's a whole other story, but it actually didn't work in my favor in the end. But of course, having the experience of writing is always very helpful. That's something I'll come to, I think, in one of your subsequent questions. So long story short, at the end of my degree, I took a course called Microbial Processes And it was really about sewage treatment, bioremediation, these ideas of where we generate types of waste products at a scale that we can't really manage through traditional ways, right? So we don't need it done in a super hurry, and we can outsource that to nature, right? So when you spill oil in a natural preserve, that can occupy huge, vast tracts of land, and we just don't have the ability to go in there and intervene. So we do things that help stimulate the microorganisms who are native to those environments to help clean those things up. So I thought that was a really powerful approach to some of these environmental questions that fit really nicely with my training in environmental toxicology. And so I started looking around for people who were doing bioremediation, and I came across a researcher in Canada at McGill who was doing bioremediation in the Arctic, which is a really interesting challenge that we still face, because if you're going to, depend on organisms in the environment to do the work in the Arctic, the work hours are really just sometimes 3 weeks, 4 weeks in the summer, right? Because it's locked in ice the rest of the year. And so we need to figure out ways to really juice that activity. And it's actually a really big problem up there because they don't have gas stations. The earth and the soil moves too much when it's freezing and thawing that they can't bury large tanks. And even when they have surface structures, they can leak, and there's actually quite a bit of spillage. So typically what happens in these very remote communities is everyone has a barrel of gasoline in their backyard and their own pump. And that's how they manage it. And of course, you can imagine how much different, there's no point source of pollution now, there's many different sources. And so it's a real issue still. And then this is maybe turning to a longer story than you want. Is this?
No, it's great. It's great. I think it's fascinating.
Okay, yeah. And so when we go up to the Arctic, I ended up studying permafrost and permafrost thaw, because that is still a major concern. But we were curious to know, are the organisms that are living in the permafrost or persisting in the permafrost upwards of, in this case where I was doing the study, tens of thousands of years, this soil had been encased in ice and below zero. What happens when that thaws? Are there still organisms that are living there that can come back to life? And what kind of activity do they have? Were they immediately going to start chewing through that carbon that we have, stored up there and release that to the atmosphere? What are the rates and what are the challenges there? And it was really, really interesting. It turns out that many of the organisms we find in the so-called active layer, the layer of soil that thaws every year, were the same as those in the permafrost. So it really was the soil itself that was dictating the survival of these organisms. And when you thawed it out, they did come back to life pretty quickly. We found, we isolated the low temperature world record holder, an organism that can reproduce at minus 16 degrees Celsius, which is basically the temperature of your freezer. So now we know that things can actually grow in your freezer, which is maybe not surprising to some people who hoard food for the apocalypse. But and this is a very special organism. I was sort of bumping up against this field called astrobiology. The study of these extreme environments where things can still eke out a living. And it was remarkable. We had isolated this organism from a saline spring that was liquid year-round. It was so salty that the water never froze. And so it was basically an underground river, some groundwater moving through. And as it moved, it picked up all sorts of salt. And when it sort of found this fissure that emerged up to the surface, it was clear water, but it made this tract of white salt. So it looked like it was snow year round. And there were things living in there, a lot of things living in there. So, minus 25 salt, about 20% salinity. So it's remarkable. But the problem with that I found was that microbes are so small and so diverse that the number of samples we could collect up in the Arctic were just never enough to satisfy a pretty rigorous scientific exploration of these things. We would come back down with 12 cores sometimes, and each one of those would be worth 3 times their weight in gold. Right? It was just to move things that far. It was 3 plane rides and 1 small twin otter. So you have limited space. You have it's all very expensive to do Arctic research, and which is sad because we do see that there's some really amazing. It's the Natural History Museum of Earth, right? There's all sorts of things locked away there that are really kind of exciting, novel, and unique. So I'd love to see more of that kind of research happen. Anyway, but I decided that I needed to go do something at a different scale. And so during my PhD, I pursued this field called stable isotope probing. And I took it to more temperate environments in forests. And I wanted to track what happens to plant matter as it sort of decomposes and enters into the soil system to sort of nourish that soil for the next generation of trees. And I did that in the context of the BC forestry industry. We actually did a continent-wide survey of different states and the forestry practices there, and sort of whether or not, the question really was whether or not if we take the trees, we cut them and we plane them in situ and leave all the branches and leaf material there, does that have a positive impact on the subsequent replanting of these trees and nourishing of the soil? And at what rate do these things occur, and which of the organisms that are important? Those kinds of questions are always being asked, because if we figure out that there are some organisms that are really, really vital to those processes, we might be able to encourage their growth in different ways. We might be able to add them back into contexts where they're not that abundant, and speed up that process. So in other words, I started working in managed ecosystems, not natural ones anymore, and starting to focus more in on, what are the processes that matter, and how can we support them, and how can we sort of, come to better understand them. And so that's what, when I started doing this stable isotope work, and that's a really powerful tool. So a stable isotope is an isotope that is not radioactive, that we can handle, we can ingest, we can work with safely, and that means we can release it into the environment without any negative consequences, and they're naturally occurring. So for example, carbon has a stable isotope called carbon-13. We're about 1% of that. 1% of our carbon on all the planet is about carbon-13, and the rest is carbon-12. And that gives us a nice hack, because we can actually artificially enrich things in carbon-13 and then track that through the environment, because anywhere that amount of carbon-13 is above that baseline natural abundance, we can say, that's from our source, and that's who's using it or who's involved in that process. So a very powerful tool that I've continued to use to this day, because it gives you a quantitative measure of things. You can say, this is how much I added, this is how much ended up in these different pools. And you can also track that into microbes, which is one of the main benefits of this technique. And it's really taking off. So the Department of Energy has now built a user facility that you can ask for them to do this, and they've automated. So they've got robots, and it's much more high throughput. And so you can see that it's really taking off because of this, the value of being able to be quantitative, and then you track that carbon. And this is kind of the amazing part. So I'll go into detail here. But that carbon, when an organism is feeding on, say, sort of glucose, let's say some sugar, you label it all carbon-13, and then whatever's feeding on that is ingesting it and making it part of their biomass, which includes their DNA. And so then we can extract that DNA from a whole community of organisms, and we can say, oh, these are the ones that are eating that sugar because they're the ones that have that 13C enrichment in their DNA. And that's a really powerful and an amazing technique to do, because it's so hard in general to know, the general lens that we look at microbial communities and do microbiome sciences is that who's all there, right? And then you just use genes to infer they're doing these functions. And we know that our ability to say for sure that that's that gene that does that function is often limited, and whether or not that organism itself is expressing those genes or actively engaging in a process that they can do is an open question. And I always love to give this example. I did a 23andMe genomes sort of test, and they recently gave me an email saying I have genes for a high performance athlete, and I can tell you that in this chair, I am no high performance athlete, but I have the genes to be one, right? So the possession of these. So stable isotope work, which is a big part of what I do, is also a way to target who's actually doing those processes. And so from there, in a way, I got sort of sucked into the stable isotope world. And so that's where I did my postdoc. That kind of was one of the key dimensions of how I ended up where I was for my postdoc at Cornell, doing someone who's also pioneering stable isotope probing work, Dan Buckley. And he works in ag. So that's another, so I've always been interested in managed ecosystems, but I hadn't done work specifically in cropping systems or, food production. And that sort of brought me into that world. And yeah, I think that's a good summary. And then so there, I just continued doing what I was doing and trying to do it better and learning. I developed some computational. So one of the things I'll talk about later is, skills that you really need to thrive in a microbiome sciences career.
Yeah.
You need some quantitative, and you need a lot of bioinformatic tools. And so machine learning is not just going to revolutionize the world, it's going to revolutionize microbiome sciences. So I dabbled in that, and I think that was really a nice experience there as well. So one of the big outcomes from that postdoc was I used standard microbiome approach, where we say who's all there. We collected about 800 to 900 samples from agricultural fields across the US, as well as a bunch of information about the properties of those soils. How rich are they in organic matter, different elements, are they, the physical structure of the soil, and those kinds of things. And I asked the question, we know that the microbes, we know there are a lot of them. We know that they do diverse things in the environment, in the soil. And are they indicative of these different properties, right? You'd imagine if soils are constantly dry because they're very sandy and the water sort of leaches through, then those organisms that live there might be more adapted to drier environments. And so how much information can we extract using these machine learning approaches to sort of predict what the conditions are in which these microbiomes exist? And it worked. I actually went into that thinking, and it should be surprising that it worked to people who are familiar with microbiome stuff, because we also know that while the microbiome is indicative of the conditions, those conditions change so rapidly and so fluctuating, and so they're very unstable. And so sometimes if you sample across the entire continent, different agricultural fields, you almost expect there to be too much background noise, too much variability to pick out a signal and say, yes, this organism corresponds to these kinds of properties. But it did work. And in some levels, in some ways, it's helped spur this interest. A lot of companies have reached out to me about this to know, like, how well does this work? And I tell them that I think there's a lot of potential there. I think the models that I developed could explain maybe upwards of 70% of the variation. So there was a strong signal there, but it wasn't going to replace the actual measurements that you would want to make. It wasn't going to say this is how much carbon you have in your soil. It's going to say, approximately within this range of error, you'll have this much carbon, but it worked. And so that's kind of fascinating. So the question now becomes, what are the better tools and approaches to sort of access and mine that information informatically to enhance those models and do a better job? And I think that's cool. And so the question is like, well, we have all these measures, why use the microbiome? And the reason is that the microbiome is integrating those signals. We make one measurement of the microbiome, all the different organisms who are responding to different chemicals, different environmental conditions, and we can extrapolate a variety of different outcomes from that. So it's a rich, what I call, information source. And I think that was one of the other key dimensions of what landed me here at Purdue, is this idea of using the microbiome for diagnostics of the environment.
I love that. I think that is so so in our future, and why I think Purdue has a great position to be able to be very impactful in that area. Rolly, the work that you just mentioned is something that I really wanted to bring up, because I've been looking at some of your publications, and I saw that publication where you talk about the health of soils in those 900 samples, and this whole concept of machine learning. Aren't we getting better? Aren't we just getting started with this? And so 70% this year could mean what next year, and what in 5 to 10 years? And what's your vision for that? Where are we going with that? And I mean, you dabbled into it. Is that where you really want to continue taking it through your time here with us at Purdue?
It's going to be a strong component of what I do. I think the challenge is really just scaling it up, right? 900 samples, already you'd expect that to not generate the most accurate models. There's just not a lot of data there, right? The models people are building from other sources like the economy and social networks, there's just millions of data points. This is just 900. So that's one of the big challenges. I think the microbiome research field has historically been somewhat divided in their methods. And so it's only in the past 5 years that we've developed more standard approaches. So really, if you go back beyond 5 years, we don't, and there's a lot of data that has accrued in that time, but it's still not enough to do some of the bigger data kinds of modeling.
Sure.
And then going forward, there's always going to be this challenge in environmental sciences because the spatial dimension, it's expensive to send people out to the field. The US did a survey of all the soils in the country back in like the turn of the century, right? We haven't really gone back. We're looking essentially for ways to not have to do that again.
Right.
But for the microbiome, I would love, France recently did this. I went out and sampled every square kilometer of the country, at least one soil sample. That kind of data set would be incredible to work with. So it's scaling to that level. I think this is where businesses come in. I can't do this work per se in my lab. I was lucky at Cornell to be situated where they have a lab that processes soils and does soil health testing. And they were an early adopter in development of that sort of pipeline for assessment. And so they were processing thousands of samples a year. So it was easy for me to sort of dip my ladle in and grab some of those. And so there are places where we can build these databases somewhat quickly, but I think the model that seems to be emerging is sort of like Facebook, where the companies can slowly get value from their product by sort of subsidizing upfront some of the costs to do the sequencing if people donate their soils and their data and those kinds of approaches. So I see a lot of value going forward there. It's just one of these awkward times where we're still learning, right? And so it's hard. These companies are struggling to know what can we actually promise and deliver on at the same time as sort of helping generate this. I guess the business model would be closer to 23andMe, right? We had the human genome, and it took decades before we actually understood concrete ways of analyzing it and working from it.
And I like the way that you phrase it, is that we're still learning. I heard this one. I'm not as afraid as artificial intelligence as I am of human stupidity, and right. That's really a lot. Are we asking the right questions? And where I was also thinking machine learning could be a big player here is, as you said, integrating the data, your background in environmental, data analysis. I mean, with the Arctic cores and trying to even look at, the chemical composition in the soils and what are the plants putting out, what are the bacteria putting out. I also see you're doing publications on understanding how bacteria are reproducing, or are they not reproducing? Are they choosing different lifestyles and different conditions? So the machine learning aspect of how can we layer that microbiome, soil microbiome data with the plant, right? The agronomic plant that is being grown on that soil, and then the atmospheric, environmental, climatic changes that are occurring, and all of this, I think would be lots of great data that could be integrated and start making a little bit more sense. And I was thinking, the time. Okay, so you took 900 core samples, but don't we have to go back and redo these and take them a meter away from where we took them? Or do we stay, and do we keep doing it repeatedly to understand how things are changing?
Yes. Yeah, that's all what it takes. And so some of it really will depend on the sequencing rates. How cheap can we get sequencing technology to enable this readily, temporally, spatially resolved data sets. And I actually do want to circle back. I mean, that was a great quote about, more for the human stupidity. And I would think that often I know that students are a little, or people who approach this machine learning question are a little afraid of it. Like I'm training this robot to replace me in a way, but I found the opposite. I mean, the models themselves are very powerful. They're a great learning tool. There are tools for the novice or intermediate person to sort of get their teeth into these things. None of it's out of your reach. And yeah, I use those things to identify organisms that are markers of different, they're not opaque, in other words. You can actually use them and play with them. And so there's a lot of fertile ground, even in this sort of monolithic idea of machine learning. There are different approaches within that that can yield interesting results.
And you're right. I mean, we're right at this exploratory new infancy stage of developing the two fields, right? Both machine learning and microbiome, especially as you pointed out, these new sequencing technologies. Can you tell us a little bit about that? What are these technologies? And I think, there'll be some people here that are listening that are thinking, okay, how do I leverage what's happening at the edge of technological development for microbiome?
Yeah, I mean, so sequencing, so there are two main technologies that people use these days. There are short read sequencing technologies that allow you to go very deep and get a lot of sequencing data, but the fragments of that DNA that you read are very short. And so when you have genes, they often exist in long stretches, and their ordering and their sort of their structure matters. So having a lot of these things doesn't always tell you the full picture. So there's been a growth in this technology called long read sequencing, which provides you longer reads, but less of a depth of sequencing. So people are combining them, of course, in different ways. And so we have both technologies. The cost per base pair sequencing back in the early 2010s was still quite high, and it has followed or actually outpaced Moore's law in terms of how cheap the sequencing has become, but it's plateaued in the last 5 years or so. So it hasn't dropped too much further, which has kind of led to bioinformaticians, biologists, microbiome researchers to start trying to eke out more from that capacity, just like you're trying to make more efficient software.
Yeah.
We're trying to do more with less. And I was quite, it's a truth about the field is that the decision for how much sequencing capacity to put towards one sample. So just for the audience, you can do one sequencing run, but you can divide that up by many different samples. You can do what's called multiplexing. So you can put one on there and that's the only thing you sequence, but you can also put hundreds of samples on there, but you get less of each.
Yep.
And so this is common practice, but we don't have a good tool to predict from, this is this sample type and this is this thing, how much of that sequencing run we need. What's the sequencing depth? So here's the best way to put it. A lot of these soil companies doing soil microbiome testing, they want to be able to say, do you have a pathogen in your soil? And how much of that is there? One of the challenges is, how deep do you have to sequence to say, deterministically that this is there or not, right? Or that these are all there or not. And there are ways to, we use other methods to say whether something's there or not that are more accurate, PCR-based methods. But ideally, like I was saying, if we could just do one sequencing run, take one measurement and answer many questions, many different pathogens, many different things, that would be the ideal outcome. So we're still struggling with that simple question of like, how do we predict what sequencing capacity we need to do the kinds of things we're doing? So I saw a really interesting poster presentation. So that's bright and new, where looking at machine learning, the different aspects of what you can get about for the property beforehand. It comes from this region. It comes from a satellite image. It has this appearance.
Yeah.
Using this a priori information, what can we predict about how deep we need to go with sequencing? So if people could get good at that, that's an interesting way of stretching.
And I think that combining that with some of your other work with looking at phenolic compounds and how these are being produced, or how they're how the microbial populations are interacting with all kinds of soil chemistries, layered on with, as you said, this genotyping or genomics analysis to understand metabolic regulation, metabolic output, who's playing with who's interacting with whom, because it must be a very dynamic interplay between so many microbes in the soil. Can you give us a little bit of an idea as to, and I know from speaking to Dr. Nakatsu, Cindy Nakatsu, for many, many years, she's helped me understand a little bit about the diversity, but tell us a little bit about that in terms of diversity, who's interacting, and this whole interplay with the chemistry and the plants and the rhizosphere. I mean, this is incredible.
You laid that out really well. Yeah, I think there's a fascination right now with coming up ways of predicting who you might find. So given these conditions, who's going to slowly take hold? And this is sort of a trade-off between sort of these chaotic things that happen, weather events. Somebody steps on your soil and, you get snakes and ladders like pushed down to the start. And so there's all these events that are outside of the control of a member of like a community that impact who ends up getting there. But there are also processes that select for organisms. So let's say a plant root is secreting these compounds that this organism loves to eat. It's just perfectly adapted to feed on these compounds. That's slowly selecting, improving the chances and that this organism will take hold in there. So modeling those processes, what we call sort of community assembly, is a very hot topic right now. And that really will influence a few things about what we're trying to do with the microbiome. We want to be able to say, we take this microbiome out of a package and we deliver it to you, and we want to say it's going to work. It's going to, the bioinoculant industry is notorious for saying these organisms, just spread them on your fields and they'll work. But we know that they don't, because they don't play well with others. That community assembly process isn't well thought through. And so that's a real question about the dynamism of a microbiome system. And then, of course, the metabolic networks. I mean, these things, I think, really, if we were to talk about the edge of where we are, the NSF just put out a call for microbial synthetic communities. So how can we put together a community of organisms that play well together, that demonstrate some sort of robust property to perturbations, to challenges that they might face in the real world? Because we've seen that one organism just doesn't cut it, right? It loses its, even if it's the most Adonis of organisms, and you throw it out into this, combat, it does well, but it slowly loses its edge. And so we need different approaches. And I think that's the sort of metabolic interdependency. An economy, essentially what it takes is a village to actually establish any one independent operating organism to perform a function. I think that's what we've slowly come to realize as an apt metaphor for that. So what are the groups of organisms that play well together, that support each other under different circumstances, and those kinds of things?
And war too, right? Who's competing against the other one, trying to produce antimicrobials that are killing the next door neighbor because they're consuming your food or whatever it might be.
It's so epic. I mean, there's so many cool, and this is really, if you're excited about some of these principles, the microbiome world, we're getting there too, to these model systems. So microfluidics, things where we can create these environments under a chip and look at them through a microscope and see some of these dynamics play out. We're at a really remarkable stage. And it can be simple too. I saw an amazing talk by a researcher who just took one of these airbrush paint sprayers, and you can adjust the fineness of the droplets. And he basically put inoculum in this sprayer, sprayed it on a slide that he could image under a microscope, and then watched what happened, because the droplets were of random sizes and of random numbers of cells. So just in this very basic experiment, you can see, what does it matter to have a small population in a big droplet or a big population, and so he could look at all of these dynamic processes. And we're still doing some of this basic research that has very profound, will have profound impacts on our understanding of how these microbiomes systems work.
And it's so neat. Again, like I want to touch on what you said, because it's so important as a way to paint the future. Lab on a chip. Let's, we're talking about what here, and how do we use it? I know you talked about microfluidics, but what are we doing here? Are we taking a clump of soil and putting it on a slide?
I think lab on a chip has a few meanings. One is that we can do, if we scale down a lot of the biochemical processes we use to study, to do analytics onto one chip, and we can do many in line. You know, as the community's living, you're able to sample different parts of it and study it as it's in its real state and in action. And then there's also sort of this idea that you can create more complex conditions than we can do normally when we grow culture. So microbiology for the longest time is you put some cells in a little tube and you shake it up and you provide the right nutrients and you try to grow them. But we know that that doesn't really make it for a lot of organisms. They have special conditions that they need to grow, and that's where they thrive. And so we're now doing a better job of creating these sort of 3D ecosystems at a small scale using things that we can still analyze and image, right? So I think that one of the cooler science fiction things that's been done recently was we use a polymer that retains elements like a soil. So it can bind cations and so forth, but it's transparent. And so and when you add water, it's perfectly optically or very close to optical transparency. So you can look right through it, and you can use high-end imaging to do a three-dimensional rastering of what's happening in this soil. So that's the next level. And what was even more amazing with that work, and I know the researcher, it was very expensive and backbreaking work. So it's proof of concept stage.
Yes.
But they can use what's called optical tweezers, which are lasers that can actually manipulate objects, and you can say, here's in this three-dimensional space where the organism is that I want to pull out, and you can fish it out of that system. So that's incredible. Yeah.
Unbelievable.
So, and I think Purdue really is situated well and to do this kind of work, right? Because we're in engineering, we have a strong engineering campus, and people are working with these kinds of technologies. We have a 3D printing hub here, so Backdown Center.
Absolutely, and in fact, I've been thinking about pulling in maybe some of the imaging people and doing an edge segment on that, and talking about some of the revolutionary image analysis that's going on here. Talk about machine learning, right? But Roland, you also pointed out the fact that we're able to start kind of probing the soil in so many different areas. One thing that is a huge interest for me is the world of sensors and biosensors. Are we going to get to the point where we'll be able to put a stake in the soil that has sensors at the end, and we'll be able to report back to the farmer, hey, either we need to add more phosphorus here, or we need to add this, collection of microbes that are going to enable these plants to access the phosphorus that's in the soil. Is this where we're going?
Yeah, that's a great example. It is, absolutely. The NSF has been putting out calls regularly to develop this technology called the Signals in Soil. I put in a proposal with a colleague here, Dr. Cliff Johnson, who's a spectroscopist, and we had some really cool ideas about how to take that to the next level. I think it's there. The challenge is cost, right? These, for a farmer, one of the challenges of that Signals program is that it needs to be scalable, right? And so, we're getting there. We can do some LED-based spectroscopy now. So we're inching along, but I think it comes down to the scalability of any one of those technologies. But there's some great ideas around that, and it's certainly necessary. It's harder in soil, so for marine researchers who do microbiome stuff, they've had it easy for a long time. They have these probes that can go up and down the column, and they can do all sorts of stuff as a lab on a chip in these situations. Soils, it's hard to dig a hole. It's a lot of work, right? So we're at a bit of a disadvantage there in those terms. But there are also people doing interesting works with it's sort of based on biological life forms and making tools like that, sort of the thing that can corkscrew through soils, that can do sensing that way. So robots that may be adapted to sensing in soil, those are on the rise. And so, I do think that that's necessary for sure. I'm not sure they must step hand in hand. I'd love to see some of those technologies growing though, because I think that would really expand my understanding of these systems.
Yeah, it'd be great. And I would think, in 5 years' time, maybe even you'd be one of those developers too, with the knowledge and the data analysis and everything that you're integrating in your lab, especially with all the engineers at Purdue, the materials engineers, the electrical engineers. I mean, there's so much of that activity as well. Roly, tell me, what is it about let's focus on soil microbiome or environmental microbiology. What is it about that field that you would want to debunk? What is it, like something that you think, this is a misconception, or this is something that we still don't know and that I really want to open up?
Yeah, we're certainly in a bit of some of the approaches that we're doing to sort of alter, in an applied sense, to use the microbiome to enhance processes in the environment that we care about. Some of that technology is overhyped, and I think that's the idea being that the microbiome can do everything. There's such a diversity of organisms, and they can do all these things. Let's just pick the right one and throw it into the environment. I think that's a very naive understanding of those challenges, and I guess what I would love to debunk is that there are so many opportunities there, but you have to think about the context a lot more. So I think one of the cooler things that's going to come of my time here at Purdue is also working with plant breeders. Yeah. So in other words, plants are the engineers in many cases of these agricultural ecosystems, of terrestrial ecosystems in general, right? And so they have a strong impact on what happens in soils, and we're pretty good at working with them. And now we have great tools to work with microbes. I think that sort of those things work hand in hand.
Yeah. Absolutely. I think that's such a great way to put it, because plants being not only the chemical engineers of the soil, but they give it structure and physical ability, and they
the highways too, along
the
roots, right?
Absolutely.
Because otherwise it's just a jumble of rocks. Imagine you walking through a jumble of rocks. It's not pleasant. But if you could just like pull along a rope, which is the plant root, you're going to get a lot further. So
absolutely. And what about the interplay between all the microbes and what we still don't know? You recently published a paper talking about the virome of soil microbes. I wonder if you could touch a little bit on what is that, and how is that adding another layer of complexity here that we still have to tackle?
It's a beautiful level of complexity though, right? Because these things we're finding viruses are so important in how organisms function. So that work was actually done with a colleague who was studying sea stars. And there was a recent sort of pandemic in sea stars that caused them to die back. And we were trying to figure out what was the virus that caused this. And we ended up finding out that it was hard to say because there were so many different viruses, preying or being part of the host eukaryotes, just like us. We're constantly being exposed to viruses that sometimes linger, they stick around with you. So what to say about viruses? So for example, these things will carry genes with them. And we know that this is one of the most amazing examples of recent years, where a virus that infects an algae that's photosynthesizing, it needs that algae to survive and long enough to produce more of those viruses. Right? If it goes in there and it's too good at killing, right, then it doesn't get anywhere. So it actually has evolved to carry a photosynthetic gene that's involved in photosynthesis to allow it to continue to generate to perform photosynthesis while the virus is festering inside that cell. And so not only is that incredible to witness, but also it makes sense now that they're shuttling genes around that confer adaptive benefits to organisms. And that might be one way of how organisms have been evolving more, to be better fit in the environment, better than just chance alone, right? And so that's the natural dimension to it. But I think, phages in general are very specific. So you can target certain organisms. So better understanding who is targeting who in the soil or in the environment will give us tools to then say, okay, if we want to target this organism, these are the phages to use. So phage therapy is groundbreaking science that's been going on for decades, but is really starting to mature. So those kind of approaches are very valuable. The problem is the viruses are a little delicate. So I think there's efforts that need to be done. Believe it or not, they don't all survive that long because they don't have the systems that a cell has to repair itself and to persist in the environment.
So fascinating. And again, like what a way to uncover again what's happening. So what you're saying is that can a virus infect a bacterium and then change its behavior or change what it's doing in the environment to then affect the plants or the soil that it's living in?
Yes, absolutely. Yeah, and that is we're really the ground zero for that kind of understanding. So the term is called a virocell. So sort of an infected cell that behaves very differently than its uninfected neighbor. So yes.
Unreal. That's awesome. And again, like to think about that world and what's happening down there, such a dynamic interplay of so many things. Let's go for a second, if you can, to the career profile. Are there things in your background that you wish you would have done more of, you would have learned more of, you would have focused more on particular areas?
Yeah, I prepared for this question because it's not an easy one. I mean, so to start out again, I was always pretty comfortable with math. And I wish I had pursued that more. So I ended up going into more chemistry and biology. But in the current stage, these things all complement each other very well. So I don't think anyone's at a disadvantage here. But having pursued, both those lines would have benefited me a lot more. I wish I could whip up a model and then say, hey, here's what I observe and here's what this model says. And I'm going that way. I'm training my students to do these things. But that's a personal blind spot that I wish I had a better background in. And then the other thing, I mean, I guess part of it is if I'm talking about microbiome, a career in microbiome science, that's one thing. If I was talking about a career in academia where I've landed, I think nothing can get done without a team, right? It's the interpersonal skills, the motivating students, and those kinds of skills are so important. And I think I'm pretty good at that, but those are kind of things I wish I started on sooner, knowing that I'd end up in a sort of a managerial role as well as a scientific role. But that might not be that useful to your audience. So, I'm not sure everyone's considering a career in academia, but yeah, for microbiome science, quantitative skills, I was able to, here's a good example. I picked up coding kind of during my master's or in the early stages of my PhD. By the end of my PhD, the funding had run out. I had published my thesis, and I was still saying to my supervisor, look, there's a really cool thing here. I want to let's do some work on here. He's like, sorry, no money, no time, time to move along. And so what I did was, because I could, source data from online public resources, and I was able to cobble together my own publication and ask and answer this question just sitting like over on weekends in my pajamas, right? So, the amount of data that's being built out there that's publicly available, because people have to publish it to, publicly to be able to put out their article. It's just incredible what kinds of questions we can answer with that data alone if you have the skills to ask those questions.
That is great. And so your coding, did you just pick up on your own out of necessity?
Yes. Yeah.
And did you take a course, or did you seek out help from online sources?
All of those things. One of the best guides out there for a biologist is a textbook called Practical Computing for Biologists. It's a great introduction to Python and all sorts of, data parsing kind of tools. I think you need a project. You need something that motivates you, that you want to sort of, push through and come up with some code that works, right? Because it's hard to learn. It's a language, right? That you really have to perfect, and it takes time. There's online resources that people should look up. There's also forums. if you can get good at posing a good question to one of these online communities, there are countless people who just love to help. It's really very welcoming community if you can sort of just figure out what's the best way to phrase this question, and somebody will come and answer for you.
Nice. And Rolly, if you were to say, what type of collaborators are you, let's say, currently looking for, or that you most frequently collaborate with because they have a complementary expertise to yours? What are those type of collaborators?
Yeah, those people like who are, in some ways I've started to branch out a lot more, because I've started to focus in on organisms that perform certain functions. An organism that degrades aromatics and phenolics, that happens in human guts, that happens in plants, that happens in the soil. So the more you learn about sort of one sort of lifestyle of organism, the more diverse kind of opportunities there are to collaborate. Traditionally, I've worked with a lot of people who are interested in, we're going to do this new management of the environment. What are the impacts? What are the things we don't necessarily expect, but we want to double check to make sure we're not polluting, or changing the microbiome in these ways. And then there are the people who are trying to model the natural systems, biogeochemists, people who are chemists by training, who focus on the environment, or physicists who are trying to, come up with the equations that best model these systems. And the microbiome is always a wrench in their works. So they prefer to make it a black box and ignore it. But nowadays, because we have these tools, they're realizing we can do more quantitative work, and we can actually build out an ecologically focused, a model. Yeah. So those are all interesting forms of collaboration. And then I guess the basic one is that's still going somewhat strong is bioprospecting. There are all sorts of interesting novel enzymes that microbes produce in the environment to catalyze reactions. Antibiotics is one classic example. And because synthetic biology, or the ability to sample a gene from the environment, put it in a workhorse organism and manufacture that ourselves it's routine science these days, it's routine technology. Going out to the environment saying, what can we discover, and what are these interesting genes? So this idea of what's called bioprospecting.
And are there companies that are starting to really go down that path because they are looking for new pharmaceuticals, as you said, antibodies, or they're interested in some materials process or chemical process?
Yeah.
And I've heard of people also looking for microbes that biosynthesize particular products that could be used as starters, raw materials for other things, right?
Absolutely. Biology does such a great job of creating variation, iterating on a theme, right? So, I saw a great talk about organisms having proto-immune systems, right? And so they have similar genes to our immune system that are evolving. And so you can maybe take something from us and evolve it in a bacteria to expose to different viruses. You can, this is not microbiome, but this is biology, and a good example. Insects have all sorts of interesting receptors in their eyes, like our retina, and they're attuned to very different frequencies of light. So you can imagine biology has found out a specific biological receptor for a whole variety of wavelengths of electromagnetic radiation. So we can look into the environment and say, we know this gene does this thing. What are all the variants of that in nature, and what do they do? So it gives us a really nice starting point sometimes to just look at, this is something I know is important for X, Y, or Z. What else is it doing in the environment, and how has it evolved?
Yeah, no, it's so fascinating. And again, you've brought up so many particular fields that are just exploding.
Yeah.
I mean, we talked about environmental stewardship and how even just the people's awareness and what's happening in the political environment now too, and how things are changing, what's happening in the global environment for stewardship, for environmental protection, and so on. But then you also talked about this advancements in bioinformatics and genomics and sequencing sciences and machine learning and all of the, the imaging lab on a chip. I mean, Rolly, this is so exciting, and what an exciting field to be a part of as it's continuing to explode this way. It's incredible.
Yeah,
I-
and it's a big tent. So there are a lot of, like, if you're interested in all these things, there's opportunity here in microbiome research. So yeah.
And if you were to say, lasting words, maybe if you remember some of your mentors that that you had as you were going through your career development, are there any parting words for people that are thinking about this field, or people that want to, maybe start getting involved?
Yeah, I do. I think two sort of sentiments to share. One is from Louis Pasteur, and the quote is, I'm sure many people are familiar with this, but in observational fields, the chance favors the prepared mind. In other words, you have to think deeply about the questions and the systems you're studying. You can't just reach out and say, I see this somewhat often because microbiome data is fairly consistent. It produces this output. And there are all these tools that you can run that output through, and it spits out a bunch of different information. And then people just present that as if it's an answer, and it's not. And so to think, to be able to look at those products, those outputs, you have to be thinking very carefully. And so it might be an accident that you discover something, but it only happens when you're really thinking deeply about these problems. And then the other thing that my postdoc supervisor, Dan Buckley, sort of mentioned when we were talking about this topic, and he said, well, really, that's an okay starting place. Like you don't have to, reject these things, but these are just the average tool. Like you go to Home Depot and you get a toolkit. It's got a hammer, it's got a screwdriver. The first thing you need to do is learn how to use these tools. But to do groundbreaking research and to advance our understanding of things, you sometimes have to develop your own tools, right? These tools aren't gonna to take you all the way that it takes to make a really complex, or not complex, but effective, let's just say efficient understanding of the world.
Yeah, and I really love the Louis Pasteur quote, because it talks not only about being prepared scientifically to encounter the surprise finding and the surprise discovery. You have to be open to allow yourself to say, okay, how do I approach this in a blank way? Fantastic. Dr. Roland Wilhelm, thank you so much for taking time to tell us about the edge of environmental microbiology and soil microbiome. Assistant Professor of Environmental Microbiology in the Department of Agronomy. Thank you again. What a fascinating conversation, and I'm going to be thinking a lot about what we spoke, and I might call you later to keep talking.
Love it. Yeah, let's keep the conversation going. And yeah, anyone in the audience who's interested, do reach out. I'm happy to respond.
Fantastic. Well, thank you again, and enjoy the rest of your day.
Awesome. You too. Bye-bye.
Bye-bye.